invite Nadira Vadaq from the Radboudumc. Please, Nadira is a postdoc working in Radboud, and she will talk about her very last published work. The floor is yours.
Yes, so good afternoon, everybody. So glad to be here. My name is Nadira Vadaq. I'm a postdoctoral researcher, a bioinformatician. I'm also a medical doctor, part of the 2000 HIV projects in the Radboudumc. So today I would like to talk more about the proteomic analysis in the context of people living with HIV. So for financial disclosures, this study is supported by Fifth Healthcare, and this is the outline of the study. So basically, we aim to identify why people living with HIV, even though they've been treated for years, having been virally suppressed, but still have an increased risk of multiple comorbidities, including cardiometabolic disease, malignancies, also HIV-associated neurology disorders. Many people think that this is caused by a chronic inflammation, persistent inflammation, also immune dysregulation in the people living with HIV in relation also with the microbial translocation and dysbiosis. And with this, we try to understand this more because the exact mechanism is still unclear using a proteomics data measured by Olink. So we use a targeted approach of 1,500 proteins with the proximity extents assay also coupled with the next generation sequencing technology. So first of all, the first objective is to actually look and to compare proteomic profile between people living with HIV and a healthy control. And next, we would like to understand better about the proteomic profile inside the people living with HIV itself. So this is the study overview. First of all, we have two different two different cohorts. The discovery and a validation cohort. The discovery cohort consisted of 205 people living with HIV and 120 healthy controls, and the validation, we have around 600 people living with HIV and 100 healthy controls. We perform a differential expression analysis between HIV and healthy controls of 1,400 proteins, and then we try to understand where these proteins possibly coming from using a tissue and cell type enrichment analysis, and also to understand the pathway that involve in the perturbations of the proteomic dysregulation. And lastly, we also try to look at the different omics. So here we integrate also a gut microbial profiling, a microbiome, plasma inflammatory markers, and comorbidities to understand the consequences of the dysregulation. First of all, we try to understand the general comparison between the people living with HIV and healthy control in the discovery and replication cohort using a principal component analysis. So as you see in the left side, that we see quite a different proteomic profile between the discovery and replication cohort of people living with HIV and a healthy control. So people living with HIV here are represented by red dots, and the black dots represented healthy controls. And then we try to find a common signature between a discovery and replication cohort using a four-quadrant plot. So in the x-axis, we see fold changes of the discovery cohort, and the y-axis is the fold changes in the validation cohort. And we can identify 276 overlap proteins that significantly differentially expressed between HIV and a healthy control, of which most of them were upregulated. And we know that these proteins, they're coming from a different Olink panels, so neurology, cardiometabolic, inflammation, and also oncology, meaning that we see like a systemic dysregulation of proteomic profile in HIV, which most of them were upregulated compared to the healthy controls. And what's interesting is also the proteins inside the circle, that we see more fatty acid binding proteins, and we know this with one of the I5, for example, is marker of microbial translocation, but also for other fatty acid binding proteins, meaning that there possibly also a gut disturbances in the people living with HIV versus healthy controls. Next, we try to understand where this protein is coming from. As we measure the proteins from the plasma, we expect that the protein is not coming from the blood cells, the immune cells, but also from other tissues. So we integrated this proteomics data with the single-cell transcriptomic publicly available data HPA and also GTAGs. We can pinpoint, for example, the possible origin of these proteins. And we find a positive enrichment, a significant enrichment of these differentially expressed proteins in the intestine, also lymphoid tissue. And from the single-cell transcriptomic, we can identify multiple gastrointestinal tract-related cells, such as proximal enterocytes, intestinal goblet cells, and pancreatic endocrine cells, but also multiple immune cells. As you can see here, we see dendritic cells, T cells, and Langerhans cells. While we try to understand the origin, we also want to know the function of these proteins, and expected. We found enrichment in the immune and lipid-related pathways. You see here, we see enrichment in the antigen presentation in the innate immune system, and then a chemokine receptor interaction, as well as the lipid metabolism. Well, we know that from the previous results, these results possibly pinpointing towards gut disturbances in the people living with HIV, we try to also understand how these proteomic profiles interact with the gut microbial species. We've also performed in the previous study a comparison between gut microbial species in the people living with HIV versus healthy control, so we know which of the microbial taxa were upregulated or downregulated in people living with HIV, and we try to also associate it with the differential expressed proteins, particularly the one that highly enrich in the intestine and lymph nodes. And we see that the enriched bacterial species in the people living with HIV, they're much more positively associated with the proteins that coming from the intestine and lymph nodes, while the depleted bacteria in people living with HIV, they're much more often to be negatively associated with the proteins that coming from the intestine and lymphoid tissues. And while understanding the biology, we're also interested to look at the possible consequences of this dysregulation. Therefore, we try to associate these proteins with the CVD history data that we have in the 2 independent cohorts. And we see, for example, around 100 or 150 proteins that's associated positively with the cardiovascular disease history. We also know for several proteins that have been known before, such as an anti-proBNP, for example. But this is with the history. We also look at the prediction model, because in the discovery cohort, we have a 5 years follow-up data of a cardiovascular disease events. And we can predict, based on these proteins, whether they are predictive or not with the cardiovascular disease events in 5-year follow-up, and we found around 100 proteins to be positively associated with the cardiovascular events. As the last resort, we try to overlap these 2 different analyses. So we try to understand first the proteins that significantly differentially express, proteins that can predict the risk of developing cardiovascular disease, also the proteins that associated with the cardiovascular disease history. And with this approach, we can pinpoint, for example, 6 proteins here that associated with the CVD. And out of these 6 proteins, 3 of them, for example, the GDF15 and Plower, they have been known to be a causative of a cardiovascular disease using a Mendelian randomization study. So at the end, the conclusions, the main findings of this project is that we know that people living with HIV on ART, they display systemic dysregulation of a proteomic profile, and these proteins mainly coming from the intestine and lymph nodes. They also enrich in the immune and lipid-related pathway, and the proteins that originated from the intestine and lymphoid tissue that much more often to be associated with the gut bacterial species. And we can also identify certain proteins that might be a targeted a drug target, for example, therapy for a cardiovascular disease in the people living with HIV. And as a conclusion, through a multi-omics approach, at least from a proteomics and microbiome, we can understand and suggest a systemic dysregulation of proteomics profile in people living with HIV, some proteins associated with the cardiovascular disease, and most of the differentially expressed proteins coming from the gut and related to a specific bacterial species. And with this, I would like to thank all of the collaborator from the 1000 HIV research project, from Radboudumc, from ETZ, Erasmus, LVG, Hanover, Ghent, and particularly of Healthcare as a funder of this study. Thank you very much.